Electron transport and ATP synthesis take place in the cell's mitochondria. Recall that mitochondria have two membranes, an inner membrane and an outer membrane. The outer membrane has channels that allow the passage of small molecules. The outer membrane is separated from the inner membrane by the intermembrane space. The inner membrane encloses the mitochondrial matrix. This membrane is impermeable to small molecules and ions. As we'll see, specific proteins are needed for transporting molecules and ions across the inner mitochondrial membrane. The electron transport chain is made up of a series of protein complexes located in the inner mitochondrial membrane. The complexes pass electrons from molecules like NADH to molecular oxygen. The electrons are transferred spontaneously when they move from molecules with low electron affinity to molecules with higher electron affinity. The electron affinity of a molecule is measured by its reduction potential, its tendency to gain electrons. Reduction potential is symbolized by capital E. Several different types of molecules are found in the electron transport chain. Flavoproteins, such as FMN and FAD, can accept one or two electrons. They make excellent bridges between two electron donors, such as NADH, and one electron acceptors. Coenzyme Q also participates in one or two electron transfers. Examples of one-electron acceptors are iron-sulfur proteins, copper-containing proteins, and cytochromes. Like hemoglobin and catalase, cytochromes contain a heme group. Heme groups have iron centers. The heme iron is converted from iron plus 2 to iron plus 3. Iron plus 2 has one more electron than iron plus 3. Recall that the TCA cycle takes place in the mitochondrial matrix. The coenzymes FAD and NAD are reduced as acetyl-CoA cycles through. The reduced coenzyme NADH transfers 2 electrons to the FMN flavoprotein, the electron acceptor, on complex 1. The electrons flow from FMN through an iron-sulfur center and are passed on to coenzyme Q. In the TCA cycle, the enzyme succinate dehydrogenase, also known as complex 2, was tightly bound to the mitochondrial inner membrane. Its coenzyme FAD accepts 2 electrons from succinate. The succinate electrons also get passed on to coenzyme Q. Coenzyme Q is a hydrophobic molecule that's membrane soluble. Coenzyme Q shuttles electrons from complexes 1 and 2 to complex 3. Complex 3 passes the electrons from coenzyme Q to cytochrome C. Unlike coenzyme Q, which is hydrophobic and membrane-soluble, cytochrome C is hydrophilic and water-soluble. Cytochrome C shuttles electrons to complex 4. Complex 4 is the last complex in the electron transport chain. In complex 4, electrons are transferred through heme and copper centers to molecular oxygen. The transfer of electrons to molecular oxygen completes the flow of electrons through the transport chain. Let's see how well we understand the relationship between the spontaneous flow of electrons and reduction potential. Use the reduction potentials of the electron transport chain components to arrange them in the order they occur in the chain. Order the electron carriers by dragging them to their relative positions in the transport chain. Position 4 is the closest to molecular oxygen. Click Submit when you're finished. Yes, that's correct. No, that's incorrect. FAD is second, coenzyme Q is third, and cytochrome C goes last. Cytochrome C has the highest potential to accept electrons. It's positioned closest to molecular oxygen in the electron transport chain. The spontaneous flow of electrons through the transport chain is coupled to the movement of protons out of the mitochondrial matrix. The protons are moved from a region of low concentration to a region of high concentration, and this requires the input of energy. Whereas some reactions are driven by the hydrolysis of ATP, the movement of protons out of the matrix is driven by electron transport. As the two electrons from NADH are transferred through complex 1, 4 protons are moved out of the matrix. 4 more protons are transported out of the matrix as 2 electrons are transferred from coenzyme Q to cytochrome C through complex 3. This mechanism is known as the Q cycle. The electrons are transferred from coenzyme Q to cytochrome C molecules. Cytochrome C delivers the electrons to complex 4. More protons are pumped out of the matrix as electrons move through complex 4. 1 proton is pumped out for every electron that enters complex 4. The ultimate electron acceptor is molecular oxygen. It takes 4 electrons and therefore 4 cytochrome C molecules to reduce 1 oxygen molecule to 2 water molecules. The electron transport chain can be disrupted. When this happens, cellular respiration stops. The compound cyanide is a well-known inhibitor of electron transport. It disrupts complex 4 and prevents electrons from reaching molecular oxygen. Cyanide poisoning is a little bit like locking the gears of a machine by inserting a metal rod. Smoking cigarettes and breathing smoke-filled air during fires are major sources of cyanide exposure. Because electron carriers cycle between oxidized and reduced forms, the blocking of electron transfer to molecular oxygen disrupts the entire transport chain. If the flow of electrons is disrupted, ATP can't be made, and aerobic organisms which depend on oxygen die.